Hello. Thank you for attending the session for Bioprocess International. My name is Kate Quigley, and I'm a product marketing lead at Benchling. It's my honor today to share some key insights we've gained at Benchling as a software provider working with leading bioprocess development teams. Today, I'll talk about how the complexities of bioprocess development can be streamlined with modern technology that increases productivity, makes high-quality data more accessible and informative, and improves collaboration within and across bioprocess teams. First, we'll talk about some common challenges and opportunities we see in how bioprocess teams work together. I'll highlight how centralizing informatics and IT infrastructure on modern software can streamline these complex workflows. I'll share introductory information on Benchling and highlight how our software has helped bioprocess development teams, many of whom are here at this conference. My primary goal will be in this presentation to inspire bioprocess teams to imagine what a more streamlined experience could look like across all of the specialized production work. First, just a short introduction to Benchling for those who aren't familiar with our company. Benchling is a cloud-based SaaS solution purpose-built to support work done in the life sciences R&D. We accelerate life science innovation by providing the modern, connected tools scientists, researchers, and developers need to succeed. Our company was founded in 2012 by a team of scientists and engineers at MIT who were frustrated by the poor quality of software they had available to support their science. Since then, our platform has been adopted by over 600 innovative life sciences companies, from cutting-edge startups through the world's largest enterprises. The biopharma industry has evolved tremendously over the past decade, and the output of the life science industry in general has been steadily increasing. We're all a part of the surge in the number of next-gen biotherapeutics entering the clinical pipeline. Benchling is proud to work with some of the leading R&D development teams across many industries within the life sciences. Let's start with some of the challenges and opportunities for increasing productivity and improving collaboration. Regardless of their size, almost all bioprocess organizations we've spoken to say that simplifying the workflows is a systems-level challenge they face acutely. One thing we've observed from our customers is that it's getting significantly more difficult to manage data and processes across the R&D lifecycle. For newer therapeutic modalities like cell and gene therapies, next-gen antibodies, and chemically modified oligos, it's difficult to track and trace the candidates, along with the plasmids and cell lines used to create them, and the experimental data that accompanies them, plus the processes used in their production and characterization. All of this means that teams spend more time looking for data, re-entering data manually, and using email and meeting time to communicate the details of these handoffs. Universally, we hear that complex handoffs of data and workflows cause drag on the overall efficiency of an R&D organization. Customers have outlined to us what an ideal system should look like across the full R&D lifecycle in order to track the scientific context and simplify collaboration throughout. Despite the overall diversity of the space, many bioprocess teams have similar goals going forward. They want to increase productivity, accelerate how quickly they can generate and use key insights, and improve collaboration overall. Customers imagine a modern organization optimized for scientific and development productivity. In this organization, they imagine their development team spending time primarily on high-value work and very little time on redundant, tedious activities. In essence, they want to help their development teams create processes with ease. At the core of that initiative is removing barriers to value-added work. Barriers are things like searching for data, manually logging experiments, which can introduce human error to data sets who have to copy and paste between systems, and reducing unneeded work. Lastly, many say that they feel they're missing out on opportunities to automate processes that could be automated. Overcoming these challenges helps free up time for the developers so they can plan new experiments and processes, interpret results, and drive innovation forward. They also want to generate key insights from how their processes run and to make informed decisions more regularly. The challenge is that the data is often spread over different ELNs, LIMS, LES, paper, disconnected on-prem solutions, spreadsheets, and even individual computers. It's prohibitively difficult to convert that raw data into something usable like reports, graphs, or just general statistical analysis. The data tends to be structured differently across the systems, making it nearly impossible to mine across samples, teams, methods, studies, processes, and programs for analytical and operational insights. And because of this, individuals may be making their own reports ad hoc to answer their own questions, but not for the broader group. Relieving this pain not only frees up time, it also makes insights more applicable to others. This accelerates the conversion of raw information into something individuals can actually do. Everything from designing and setting up processes, deciding what candidate to advance forward further. Bioprocess development team is a team sport, now more than ever. How well developers work together unlocks not only their individual capabilities, but compounds the overall potential for their collective efforts. The challenge, however, is ensuring that everyone communicates and works together effectively. If people don't speak the same language or don't have the entire picture of a project, they may give uninformed or inaccurate guidance. Data needs to be consistent, high quality, and reliable over time, including in real time, in order to be useful. We all know how much is sunk in handoffs between the many meetings and copying information from one source to another. It's also typically quite challenging to sort out who is working on what and when that work might be done. A lot of time is wasted spending on organizing tasks. Improving your IT landscape is a tangible step forward. So let's take a look at what you should be doing in order to make that happen. Bioprocess development teams are clearly seeking a single interface to model how their organizations work, be it within their teams, across divisions, or amongst sites. They want to increase accessibility of high quality data and reduce or eliminate point solutions altogether. Of particular interest is to have software that makes tech transfer from discovery to development more manageable. IT teams want solutions that are easily connected with other solutions to structure data with consistency across the organization. They also want the solutions to be highly configurable so they don't have to stitch it all together with custom integrations and code. Everyone also wants solutions that evolve in order to keep a pace of the innovations happening within the company. These updates should be managed by the platforms themselves to reduce the total cost of ownership. To that end, Benchling customers can now license a single unified system for R&D that has been historically served by disparate ELN, LIMS, and LES software. Modern electronic lab notebooks should not only document experiments, they should also provide process and sample management. LIMS and LES should have experimental context and biological understanding in addition to core sample traceability. Although processes need to be locked down for regulatory and compliance reasons, solutions need configurability that adapts to process changes. Improving your bioprocess development to accelerate time to market is much more feasible within Benchling. Benchling is a modern, unified, and easy-to-use R&D cloud software that allows scientists to seamlessly move from research through development within a single system. The R&D cloud is designed to accommodate both the flexibility and speed needed for bioprocess development, as well as the control and compliance required for regulated processes. The platform is experimentally driven, sample-driven, and procedurally driven. Benchling supports the distinct needs of those focused on clinical and commercial development by hosting the applications in Benchling Validated Cloud, an environment for those within compliance requirements. Our quality team helps our customers in Validated Cloud manage their validation cycles with ongoing support, including installation and operational qualifications guides. With continuous innovation, we increasingly support our customers who are using us in bioprocess development. These teams use our solution in environments validated for GLP, GCLP, and into phase 1 GMP controls. The solution represents fundamental building blocks for bringing products towards the point of clinical evaluation and manufacturing scale-up within Benchling as quickly as possible. This digital solution builds upon the capability typically found in traditional ELN, LIMS, and LES, while providing a unified and intuitive solution that adapts to meet the evolving needs of process development teams. Bioprocess teams require flexibility from their tech solutions as they explore parameters during process characterization. As the parameters are tested and narrowed down, however, they need to execute experiments repeatedly to test process reliability. During this transition, Benchling's ability to move fluidly between flexible work into structuring repeatable tasks becomes quite powerful. These teams can make this pivot by creating and using controlled templates and results tables. The same functionality supports further process refinements as needed to increase experimental velocity. Having all bioprocess development teams on Benchling makes data sharing and sample tracking manageable. By orchestrating tasks, work runs smoothly across teams. Handoffs always provide context of prior experimentation. The data set is centralized, standardized, and can be easily aggregated. This makes it easier to generate insights in dashboards and create key reports across samples, teams, methods, studies, processes, and programs. As an outcome of Benchling solutions, customers reveal that their time spent on average is 64% less just on requesting submissions and tracking statuses. They also report a 46% improvement in ease of collaboration and sharing data. As a next step, we'd like to share stories. We've got one upcoming, and we'd still love to have more conversations about other customers who have had success using our bioprocess development solution. Some of these stories show how your organization can achieve similar gains in productivity and data quality, sharing insights and advancing programs more efficiently. We're eager to learn more about your organization's needs and share in the success collectively. With that, I would like to show a recent interview we did with Dr. Zora Merizaga-Vivienne and Samantha Povlech and what they've accomplished with their teams at Q Biopharma.
We are all about harnessing the power of the immune system to develop therapeutics for either cancer or autoimmune diseases.
When the company was founded, in the immunology part, we were 2 scientists. Q had a very basic notebook system back at the time, and we understood very quickly that we needed something more powerful for our research if we want to go to the clinic, if we want to be public, if we just want to evolve on the right direction.
We are trying to develop therapeutics for patient populations, especially those that may not have responded to existing therapies on the market. We are trying to make sure that the products that do end up on the market that can help these subset of patients are both safe and efficacious. I've worked in early discovery, I've worked in process development, I've worked in late stage development, CMC. There is a flow of information back and forth that should be there. And Benchling allows us to have that flow of information back and forth instead of a unidirectional information exchange. So as many organizations go through a process of both research and development, there's usually a transition point. A lot of times you have the research side basically tossing something over the fence to the development team, and then they take it and run with it, and it's gone. So if you have your researchers think about how they're collecting data, what data they're collecting in that way, it's a little bit easier to push things into the development space where you may not have to redo experiments over, those early experiments over, because it's already been captured by the research group in a way that is acceptable to regulatory agencies. What Benchling has allowed for is that back and forth. We can bring on our own process development in-house now, and so there's a bit more conversation back and forth between our research organization and our process development function, which, honestly, it gives us better tools for research.
We need standardization to make sure everybody does a process the same way. So everything is repeatable. It doesn't matter which technician is doing it. We can trust the product they give us, which is the most important at the end, because the data we're generating in our lab will be used to ask for an IND, so to ask authorizations to test these molecules in humans. So of course, everything has to be standardized, optimized, and kind of perfect. So it helped us to just save so much time and energy doing everything at the same time. Having softwares like Benchling, so it's a LIMS system on top of an inventory system, on top of a notebook system, it helped us to organize all this.
Most people have had cancer affect someone in their lives at some point. It is nice to be part of a potential solution to a lot of problems.
It's such an illuminating story of what modern teamwork looks like. We're fans of the incredible work that Q has been doing on Benchling. With that, I'd like to thank you for joining me today. To discuss your team's collaboration needs, please reach out directly if you'd like to learn more about Benchling. My email address is here at kate.quigley, Quigley, @benchling.com. And we'd also love to see you in our virtual booth. Thanks, and have a great day.